So kia ora everybody. My name's Sally Ann Harbison, and for those of you who don't know me, and I think that's probably quite a few, I'm from the Forensic Biology Laboratory in Auckland in New Zealand. So I guess I came the furthest. Obviously we've all heard already that research and development into the application of MPS technology is proceeding at a fast pace, and at ESR that's no exception. So we've embarked on a research and evaluation program, and our focus is really on transforming our forensic workflows to MPS. So in this presentation, I'm just going to talk about some of the ways that we're going about this and use some of our experiences from our research program to highlight some of the key areas, or what we think are the key areas. And one of the big things that several people have already mentioned is it's not just about the technology, but it's also about the people who are going to have to do this in our laboratories. So first of all, what is it that we're wanting to replace? So we're a national laboratory. I used the map because lots of people don't know where we are. So we're the little comma down the bottom here, off the coast of Australia. We're the sole provider to the New Zealand Police, and we operate the National DNA Databank. So at the moment we're using 4 of the STR multiplexes, which are up here, and we're using continuous models for DNA interpretation, and Bruce very kindly mentioned StarMix this morning. So I can talk to you about that if you're interested. We're still doing 34-cycle SGM plus low copy number testing. We do RNA testing for body fluid ID, and we use laser microdissection. So I'm pretty sure we're all agreed anyway, that so far our existing platforms are very sensitive, they're very well characterized, we know how they work, we know what to do when they don't work, we have methods to interpret the data. I think we're all more or less doing the same thing. We've been doing it for a long time now. STR's been around for ages. Our turnaround times are really fast. So, I mean, I'm from an operational laboratory, and we would not 1 or 2 days is par for the course, really. And the evidence is generally accepted in court with the odd hiccup here and there. So we're looking for something that's going to be robust and well characterized and supported by you guys in the community. And one of the good things about a day like today, when we all sort of stand up and say more or less the same thing, is that kind of reinforces that this is a technology that is supported in the community. And that gives me a great deal of confidence going forward. We need to be compatible with our data bank. I'm not intending to replace it at the moment, but we need to be compatible with it. We need the same or similar levels of sensitivity, but we need something extra, right? So we need some more information for every sample. We're going to need some bioinformatics, and we really do need to be able to churn our cases out on average in 5 days, or samples out on average in 5 days. And then this is the lab management thing at the bottom, cost neutral, not only in terms of reagents, but also in terms of people's time. So we started our research project 3 years ago, and we had 3 objectives. And I'm going to talk today about the first 2 of these. So we also have a non-human DNA testing project going on as well, which is primarily species identification from mixed samples. And I've got a couple of slides right at the end about that. But I'm going to focus on the first 2 today. So we're from New Zealand, and we do things a bit differently sometimes. We are a long way away. Maybe we don't hear what you guys are up to some of the time. The first thing we wanted to know was whether or not we could sequence all the samples we already have that we've already amplified with identifier and PPY and are in the freezer. So that was the first objective. And then at the same time, we've designed and tested our own custom panel of markers. And I'm not going to dwell on how you do this thing, because I think most of us know more or less how you do this. And this first slide is basically extract, quant, and amp, as per normal, with a few extra purification and quantification steps in there. And we had to use the Kappa Ion Torrent DNA library prep kit for this because we already had amplified DNA fragments with fluorophores on, and yeah, it does work. And then a very traditional PGM workflow, including the various quantitation and quality steps. For the work I'm talking about today, we used the OneTouch 2 400 base pair kit because the Ion Chef kit wasn't available at the time. And this particular piece of work was on the 316 chip. So obviously that initial QC is carried out on the Torrent server, but we really want to be able to interrogate this data ourselves. So we do a lot of our own bioinformatics, and we have a little pipeline that we've set up for this. And we're using a lot of open source software for that, which is something that forensic labs aren't very keen on, but that's what we're doing. And we're using, for alignment, we're using Bowtie 2 and Straight Razor. And at the time we did this particular project, we were using version 1.2, which has since been superseded. So just sort of touching on a few of the results in some of the areas I think that we have some improvement. So 3/4 of the reads were able to be aligned with an STR sequence of some sort, and that was with Bowtie 2. But only 1/3 of those were able to be aligned with Straight Razor, so that means they span the entire repeat region. And clearly maximising the length of our sequencing of our Amplicons is really important for us, and that's something that we're working on at the moment. We needed to start with some analysis guidelines, and yes, this is binary, sorry, not continuous. But similar things to what I've heard people talking about this morning. So basically we're just using our old CE thresholds from years gone by to interpret our data as the first point of call, and it works just fine. So locus success rates are reasonable, but not brilliant. Some profiles are full, some aren't. Most of the partials are FGA. The PPY loci are much more successful than the identifier ones. And 3892 is where all the no results come from PPY. So that's a primer design issue for us, I think. Some observations which you've already seen people talking about today. And we haven't used the Haiku kit yet because it wasn't available to us at this time we did this work. So we're really hoping that that will overcome the homopolymer region in FGA, because FGA is the one locus that's not very good for us at the moment. And what we've done in the interim is we group, for example, this is an FGA 20 allele, we would group the 19.3 and the 20 together in order to carry on with the analysis. And like everybody else, heaps of sequence variation, so I'll just go past that one. So remember, this is a sample's already amplified with identifier or PPY and then sequenced. So in the grey, you can see the RFU for identifier, and then in the brown, the mapped reads for the STR loci. And THO and TPOX, and we'll see this again later, are always the ones that for us amplify more than the others. This is organised by Amplicon length, so not an Amplicon length issue and not a repeat sequence structure issue either. We did have some mixtures, and I just put up a couple of examples. So applying our old-fashioned CE binary rules is reasonably straightforward to interpret a mixture if you've got enough reads, using exactly the same criteria that we would have used previously. And then when you don't have enough reads, as in this case, the 27 has dropped out, and given that we know what it is, we know we have a partial profile. So the validation and the improvements are all around this kind of area, the interpretation of the results. So we also have our own custom primer panel, which we made ourselves. It has 67 STRs and 211 SNPs plus aminogenin in it. And again, the workflow is pretty similar, actually, especially once you get to the Ion Torrent. Ampli-seq library prep, in normal PGM workflow, but with a 318 chip this time. And the bioinformatics was exactly the same. And the same result was obtained from the mapped reads, so 76% of them aligned to an STR or a SNP sequence with bow tie 2, but only a small proportion of those, a much smaller proportion of those, were able to be aligned with straight razor, whereas almost all of the SNP ones were. And this is a similar slide to the previous one. Really the only thing to point out is that it wasn't so good as the previous study. It's hardly surprising given the number of markers, and it's not been very well, we haven't optimized it at all yet. And in some samples, almost all the SNPs typed, and in others, not all of them. I just wanted to pop this one up. This is again the same, so on this side is the same imbalance between loci slide that I showed before, and on the left, on the extreme left is the same identifier loci in the custom primer panel, organised by the size that they are in that panel, and then in the middle, the size that they would be in identifier. And again, THO and TPOX have stood out for us as the loci that need the most work in terms of imbalancing, and yet these are two completely different sets of primers that have been used here. So wasn't really a surprise to an old STR person like me that THO would be like that. I kind of guess, if I had to guess, that's the one I would have picked. So one of our YSTR loci and 5 of our SNPs didn't work at all. And then some of the others either amplifying off the target or amplifying within the target and not covering the full repeat. And so, interestingly, a lot of those are YSTRs. I'm not sure why, but primer design is being redone for those to see if we can make that better. So the main purpose of me wanting to talk today was to talk about implementation, and if those of you are 1785 audited will recognise some of the titles here, because I picked out some of the ones that I think are key for us going forward. So there are already guidelines and standards for MPS and clinical work. This is from the Royal College of Australian Pathologists, I think, or Royal College of Pathologists Australia, I think. And these guidelines are there, out there, and you can access them, and they're very similar to what we would expect, but I guess my point is that other disciplines already have them, so I think it would be sensible for us to have a look and see what they say. And if anybody really wants to read them, I have a copy in my room, which I'd be happy to pass on. The biggest thing for me as a sort of technical manager is our personnel and their qualifications and training. We do need different skill sets, especially in this initial stage of evaluation and development. And we do need access to and employment of bioinformaticians, which we have. You will need some statisticians, which we have. And if you've already got them, you need to direct them down a different path, which is not always easy. Special people to run your sequences. This is a very operational focused talk, you probably picked up by now. One of the problems that we have with this rapidly evolving field is the change in technology and the new kits that keep coming out and the new software that keeps coming out. And if you've developed yourself a nice little validation plan and then somebody brings out a new kit and then you have to start again, not good. So having a good relationship with your suppliers and knowing what's coming up in advance is really important. So we've also engaged in a lot of training. So we've been training our own biology staff. Not the ones involved in the project, there are about 10 of us involved in the project, but the rest, the other 30 odd, are already embarking on a training program which starts with familiarisation with all the different words and the different language and the different techniques, and will eventually result in them reporting their own casework in court. We've also been talking with our police, we've been talking with our managers who need to fund us, and we're about to start with a community engagement, talking with the public about what this new technology will offer in the future. And I think that's really important because there is so much more that we're going to be able to do. The public need not to be frightened of it. If we don't tell them in advance, then we run the risk of them kind of throwing their toys out of the cot and saying, no, you can't do it. I think lab design probably isn't really necessary, to be honest, but the work areas are all the same. And Bruce talked about the kind of process of sequencing a sample being very similar to what we're currently doing, but exactly the same in your lab design. Your work areas will be the same. The red lines are where we have separate rooms for the different tasks that we're doing. And so there's no need to engage in new buildings or anything of that sort. So selecting what you're going to do is going to be quite important, I think, especially if you're not in the research space particularly. There are different sequencing chemistries, and some people touched on some of those today, and they have pros and cons, just like the PGM does. And there's a balance that you need to establish in your own minds about which of the chemistries that you think is going to work best in your situation. Obviously, there are community panels now, which you might be wanting to use, and if you have no R&D, that's probably a good option, or else you can make your own. You might want to do everything all at once, or you might just want to do what you need to do for your particular case. I'm kind of veering to the everything all at once, but it might be too hard in the short term. The biggest problem for us all is going to be understanding the stochastic variation that's going on in these reactions, and there are some extra steps depending on which technology you decide to use, and so each of those steps is going to have an influence on the results, and you need to understand them. And I think you need to be able to do the bioinformatics yourself, or at least somebody does, in order to understand them properly and whilst you're doing your evaluation and validation. Okay. Another big one from a management perspective is what you do with the data. At the moment, we're keeping everything, but obviously you can't. That's not feasible in the long run. So we all need to decide, and we probably should do that as a group somehow, what data we're going to keep and how long for and where, and at what stage are we going to keep that data. At the moment, we have hard drives stacked up in my office with each experiment on them. That's clearly not sustainable in a delivery sense. And as a security kind of organisation, we can't, at the moment, store stuff in the cloud, but I think that's going to have to come. I don't see that there's any real option there. There's going to have to be some server system somewhere to store all this stuff. And then what do we do about the incidental findings? So we might not think we're going to find any because we're using targeted PCR mostly, but we will for sure. We don't know enough about all these markers yet to know whether or not we're going to find anything that we might not want to know. So we have to have some kind of plan around that. I'll skip through this because I think this is a bit self-evident really, but there's so many options out there if you want to do it yourself. But most of them are similar, and most of them are the open source command line programs. They're really easy to do. I can do it. If I can script stuff, so can you. It's not that hard. Somebody shows you what to do, you can do it. We're using Straight Razor for STR alignment. We found it really easy to use. Thank you, Bruce. And it's easy to edit and modify yourself as well, so you can add in your own versions of things if you want to. So that's been really good, and we're really happy with it. I was in two minds about this slide. Haven't decided yet what the controls are going to be, and I'm not talking about test fragments and the controls that are sort of built into the sequencing process, but the standard controls that we've always used, that E positives and extraction negatives and AMP positives and AMP negatives. Exactly how many of those we're going to end up with, I'm not sure. We've started off with most of them, but I think we might drop a few as we go. And we are, at the moment, we're using NIST standards as well as our in-house controls for that. Because I'm old school, I like to see stuff, and so I'm a big fan of the Bioanalyzer. I'm not sure how many of you have used a Bioanalyzer, but it's like a mini gel on speed. And it's really nice to be able to see your Amplicons. I know you can't pick out each individual one, but at least you get an idea about the range and the success of your amplifications that you're going through the different steps. And there's lots of different options for measuring how much you've got of the various parts of the process as well. Okay. So I mentioned before that we have to start somewhere with some allele calling. So these were the guidelines that we came up with. And this is really where we're putting the effort at the moment in terms of our validation. So I haven't got as much data as everybody else. I'm a bit embarrassed, to be honest. But anyway, we've plotted our imbalance for all of the data that we have, and it kind of fits the expected little shape that we were looking for. At the moment, we need quite a few reads to get to a 50% imbalance, but it's something to start from and something to be improved on. So what are we actually aiming to do? Well, we did think we would start some sort of specialist service in a couple of years from now. That's what we're aiming for. Another year of validation and another year of development, and then a year of validation, more or less. And I'm hopeful that we will be able to do everything in a 3 to 5 year period, our casework samples, all in one platform. Because as a manager, I do not want multiple platforms if I can avoid it. Okay. In terms of our development, obviously, we need a stable platform. So we're adjusting the method, we're adjusting the multiplexes, we're adjusting the primers, we're tinkering with library prep and templating and all those sorts of things. I'm looking to get a stable platform to proceed a validation with. Because we're ESR, we like mixtures, so we do lots of mixture studies, and that's one of the sort of main focuses for some of the casework people on our team. We do need to be developing our software for compatibility with the databank, but I don't see that as a big issue. We are looking at ethical concerns and policy issues. We have to figure out what we're going to do about population data, because we haven't used most of these markers before, and we're going to need to access them from the online community somehow. So we need to figure out a way of doing that makes sense for us, and lots more training. On the research side of things, and we've just started our first ever MITO project. We've never done MITO before, so we've just started that, and that will be with a focus on hairs. And we've been running a twins project for the last year. We've been looking at microbial community profiling. ESR has an infectious disease group who do a lot of microbial genetics, and they've been helping us with that. And we've also been doing some species identification. And again, we have a food safety group at ESR, and so we've been working with them on some of the food forensic studies. And please don't ask me what's in those sausages. It's just not nice. So I'd just like to finish by just saying that I think this meeting has probably reinforced my belief that this is a suitable technology for us, and it's already being widely used in the clinical field. It's in the capability of all of you, providing that you invest in sufficient training and resources. I think if you're not doing R&D, then you've got a good community solution out there you can adopt. Maybe not tomorrow, but in a year or so. And if you really want to, you can go and do your own thing, and you'll get great support from all the suppliers to do that as well. So it's at the moment, it's very flexible. It's well worth having a dig around in the bioinformatics. And I think we're all going there, aren't we? So just to finish by acknowledging Ryan, who does almost all of the lab work on this and most of the data analysis. New Zealand Genomics is a New Zealand government infrastructure provider, and they did the initial sequencing for us and helped us get the bioinformatics underway. I had the job of teaching me how to write my own scripts. The New Zealand government has paid for all of this research through ESR, and I would just like to thank Life Technologies, but now I can call you Applied Biosystems again. I'm so excited. And Lucy and Beatrix, who are here somewhere, for assisting with all the custom primer panel design, and also with inviting me to come along and talk to you today. Thank you.